us. Okay, and we are live. Got it. Okay, let's see. We've got some participants trickling in. I'm going to give it another minute and get a little prepared on my end. So you've been in Flagstaff since 2015?
2014, yep.
2014, okay.
A little longer, but not much. Yeah.
Had you been exposed to the region beforehand, or was it kind of a new thing?
It was kind of a new thing. So I'm from the West, I'm from Oregon. So when my wife and I were searching for places to go, we wanted to come back West. But I had not spent a lot of time in Northern Arizona at all.
Okay. But I mean, coming from Oregon, you were pretty close here.
Yeah, we definitely appreciate being back West.
Yeah, you still have family back in Oregon. You travel up north a little bit.
We do, yeah.
Where in Oregon are you from?
Corvallis is where I grew up.
Oh, okay. I have Oregon is still on my list of places to visit. It was actually our planned vacation for summer of 2020, and we all know how.
Yeah, that got disrupted. Well, yeah, you absolutely should. Yeah, it's a wonderful place.
And are you doing research at NAU, or are you instructing? I know you're instructing, but are you doing research as well? Are you going to talk about that tonight, or?
Yep. So my talk tonight is about research that I've been doing with students here, and then some collaborators at Lowell and various other places.
Okay, then I definitely don't want to spoil anything for the folks that are joining us. I'm going to go ahead and get started, Garrick, if you don't mind.
Great.
Welcome everybody tonight. Thank you for attending this NAU VPR Innovation Series lecture. My name is Joe Wagner. I am the Flagstaff Festival of Science Treasurer. Joining me tonight is another board member, Dave Gillette, and also our speaker, Garrick Lindbergh, who's here to present on the topic. Before we get started, I would like all of you to know that the festival is live streaming this webinar to YouTube. Only the hosts and panelists will be visible or heard during the simulcast. This event tonight is part of the Flagstaff Festival of Science, which are the best 10 days of the year. Our mission is to connect and inspire the citizens of Northern Arizona, particularly youth, with the wonders of science and the joy of scientific discovery. Your participation and support are vital to our shared success. All festival events are free, thanks to our generous sponsors and donors, which we'll show you at the end here. And if you would like to support our work around the community, please go to scifest.org and click donate. This is only one out of nearly 100 events during the festival. You can find a full list at scifest.org. Some events are pre-ordered tickets, and all of our webinars offer the ability to pre-register for admission. The Flagstaff's Eventbrite page, flgscifest.eventbrite.com, has all of our webinar registration links and many of the tickets for events that require a reservation. On the festival's webpage, scifest.org, in addition, you can find a listing of all events and links to our YouTube page, where you can find recorded versions of these webinars and talks from this festival and previous festivals. Links to the festival's website and Eventbrite can be found in the chat. I will post that over right now. If you'll all give me a moment here. Okay, moving on here. If you missed parts of this stream or any other festival events like the keynote speech that was last Friday night, you can go to the Flagstaff Festival of Science YouTube page and watch a replay. A couple upcoming events that I am still looking forward to. We have our Tag a Monarch repeat on Saturday. It happened last Sunday, and we do it again this weekend. And the Lowell Observatory Star Parties that happen on Thursday, Friday, and Saturday of next weekend, which will wrap up our 2021 festival. During the presentation, please type your questions in the Q&A box. If you see a question you like, please upvote it. Due to possible time constraints, we may prioritize the questions that have upvotes. If you have technical problems with the webinar, please communicate to us in the chat and not the Q&A box. One other way you can support the festival is to take a short 5-minute survey after the completion of this webinar and any other events that you participate in. The data is very helpful for the board to improve, as well as a great source of data for our grant applications, which keeps everything free for everyone. The survey can be found in the chat. I posted it a few minutes ago, and one survey respondent will win a pair of Apple AirPods after the completion of the festival. So please fill out the survey so we can get your feedback. All right, thanks for patiently waiting for the housekeeping portion of this presentation. You're probably excited, so I will introduce our speaker. Garrick Lindbergh grew up in Oregon and earned his undergraduate degree from Oregon State University in chemistry and mathematics. While at Oregon State, he worked in toxicology lab, which made him aware of the possibilities of doing science and working in a lab, and set him on the track to become a scientist. After graduating from Oregon State in 2005, he traveled across the country to earn a PhD in physical chemistry at Boston University. His graduate work centered on developing theoretical methods to understand phase transitions. After earning his PhD in 2011, he became a postdoc at the University of Chicago, working to design more robust membranes for use in clean electrochemical devices. In 2014, just recently, joined the Northern Arizona University as an assistant professor, and in 2020, he was promoted to associate professor of chemical physics. Garrick tonight is here to talk about searching for life in the rivers and lakes of the outer solar system. Garrick, go ahead and take it away.
Great. Thank you, Joe. Thank you, everyone, for taking time out of your evening to listen to me. Hopefully you find this interesting. So yeah, I want to talk about some work that I've been doing over the last 5 or so years, trying to understand the solar system, and in particular trying to connect that to how we understand where life might have come from. A lot of this work grew out of the New Horizons mission, and in particular, some friendships, collaborations that I've developed with 2 planetary scientists at Lowell, Jennifer Hanley and Will Grundy. And I met them early on in my time in Flagstaff, and they introduced me in kind of more depth than I would have casually gotten to the New Horizons mission and the amazing results that we've gotten back from it. So you're probably aware of this. You're living in Flagstaff. Many of you are living in Flagstaff at least. But New Horizons is this probe that we launched that has gone out into the very deep depths of the solar system, and it's sent back all this amazing information about Pluto and other places. And so this was kind of a jumping off point for me to get involved with some of these projects. If you get nothing else out of this project, out of my talk, I think just taking a little bit of time to go look at some of these photos that we got because of New Horizons is well worth your time. So if we go back to some of the best pictures we had before New Horizons, we have this blurry blob on the left. On the right, we have one of the photos that came from New Horizons. So it really gave us unbelievable insight into what this object looks like way out there. And if you just flip through some of these pictures, it's so spectacular being able to imagine yourself actually on Pluto. I find this, I don't know, humbling and inspiring. So I'm just going to flip through a few of these and try to transition to the motivation that inspired some of the work we've done. So for example, here you see these ice flows kind of in the foreground, and then behind it, them running up into these mountains on Pluto's surface. Here we see a zoomed in portion on some of these ice flows, and just the texture on this, the complexity of this is, I think, different than what I would have imagined before I started flipping through these pictures at least. Yeah, if you zoom in, there's lots of colors that come out on these. So there's different molecules that end up being laid across the surface in different patterns. There's all kinds of different structures that emerge as these ice flows do what they do across the surface. In addition to that, there's an atmosphere there. We can actually look at the atmosphere and you can see that there are gases floating around Pluto's surface that contribute to not only there being interesting surface processes, but something sitting above that. And so what I want to do is try to take this new information that we're getting from Pluto, from the New Horizons probe, and connect it to how can we start thinking about where are other places in the solar system that maybe life could form. Oftentimes when you hear this idea get presented, it'll be presented in terms of Earth analogs. Absolutely, that makes sense. We understand life on Earth better than anywhere else. But I want to broaden this a little bit to think about how could maybe other places in the solar system be a situation that we could also use to understand the formation of life. So instead of using Earth analog as our motivation, I want to kind of think a little more broadly. And the way that I'm going to do that is say that we definitely need to have energy and molecules around in order to form complex molecules. That seems to be central to the formation of life. And in addition to that, it seems like life really needs liquids. Oftentimes you'll see this limited to liquid water, but I want to think a little bit more broadly. If we have liquids, then you can imagine these molecules wandering around, bumping into each other, and potentially doing the processes that might lead to the formation of life. So the structure of my talk is going to be kind of targeting these two different points. I want to look for where are liquids in the solar system, where could they possibly be that we haven't found them yet? And then given that we know they're in some places, what could be happening in them? And then finally, what happens with these complex molecules? Can they behave the way that the molecules of life behave? So I guess I got a little bit ahead of myself, but these are the main questions that I'm going to target. And I have three different kinds of threads of work that my group has worked on that I'm going to try and weave together to hopefully tell a story of how we are identifying places in the solar system where these sorts of interesting chemistries could be happening. Okay, so back to pictures of Pluto. These are probably the most interesting part of my talk. Here we have some zoomed in photos of Pluto. And what's interesting, what these arrows are trying to highlight are locations where the structures that we see look like they could be caused by the erosive action of liquids. So these look like maybe they could have been some sort of maybe transient formation of liquids that then moved across the surface and caused it to have these patterns. This is kind of tantalizing. We don't have direct evidence. We don't see the sloshing around directly, but maybe these are indications of something interesting happening. There's other evidence of this as well. If you zoom in on regions, there's stuff floating around Pluto. There's other things in space around it. Those things will eventually collide with Pluto. But if you look at its surface, it does not have as many craters as you would expect, given the density of objects floating around it, which is another piece of evidence that maybe there's something more going on to cause resurfacing on the surface. One really clean way to see this is if you look at Pluto's moon, Charon, and compare it with that. There are far more craters on the surface of Charon than there are on Pluto, which gives credence to this. I'm not going to say that liquids are definitely the cause of this, but they are one possible explanation that is kind of an interesting thing to think about happening on the surface of Pluto. What's kind of strange about this, though, is that if you look at the molecules that are present there, and we have a very good inventory of what molecules are there because New Horizons had spectroscopic instruments that allowed us to look at what's there, all of these molecules should be solid at this point. So it's primarily, there's a lot of nitrogen there. There's a lot of methane and carbon monoxide and ethane, smaller amounts of larger hydrocarbons. Most of these things you would expect to be solid here. So if there are liquids there, you kind of have to start wondering how would they actually come about. And so at the bottom here, I give the temperatures that we're thinking about. So we have a range of about 30 to 60 Kelvin are common temperatures on the surface of Pluto. And if we just start picking apart these molecules, all of them are solid at these temperatures, the temperatures that we would think are important for Pluto. So how can we go about explaining what could be going on here? Well, we have one common example of where you take two things and mix them together and you end up getting kind of very different thermodynamic behavior on Earth that we're all pretty familiar with. So when it gets cold here, I guess we don't do this in Flagstaff because we don't salt the roads, but we're all familiar with the idea of when it starts getting cold, there'll be trucks driving around dropping salt onto the road because that will cause the freezing point of water to drop a little bit. This general idea in thermodynamics is something called eutectic behavior. So eutectic means easy melting in Greek, and it is just the idea that sometimes when you take things together, you take things and mix them together, you can end up with a mixture that has lower melting points than any of the individual species that you put in there to begin with. So if you have this sort of phenomena going on there, we could have mixtures of molecules that all should be solids in their pure forms at the temperatures that we see on Pluto. But maybe when we mix them together, we start to see them forming liquids in the conditions that occur on Pluto's surface. And so here is a general kind of form of the way that we oftentimes will represent this in thermodynamics. At the two sides of this plot, so where you see the vertical black lines, those would be pure molecule behavior. And then as you mix them together, you see the blue line predicts decreasing melting behavior at every composition. There is some limit to this. In the middle, this reaches this kind of vertex, inverted vertex, where we have a minimum. And so that is our maximum melting point depression that we can get from this sort of behavior. And so the first thing that my group has been doing is exploring this sort of eutectic behavior given the types of materials that we have on Pluto. And so in order to attack this, we developed a relatively simple thermodynamic model. If you remember your physical chemistry at all, it's based upon colligative property behavior. And from this, we can predict this freezing point depression, and we can predict that maximum freezing point depression point. And so here is an example where we did this with a mixture of methane and ethane. And what's interesting, what's kind of validating about this, is here at NAU, we also have a pretty sophisticated apparatus that allows some of my collaborators, you can see them in the picture there in the corner, to actually mix these sorts of things together and observe what actually happens. So all of this intense machinery, which kind of scares me as a theorist, all of this comes together to allow them to actually mix methane and ethane in there, for example, and cool it all the way down to these temperatures that are relevant for understanding the surface of Pluto. They did experiments like this with methane and ethane, and here's what they found in blue. The black lines are the data that I just showed you a few slides ago from our thermodynamic model. So this grants some credence to maybe our simple model allows us to do what's really difficult to measure from the probe and even difficult to do in the lab and come up with predictions for how could you actually get liquids under these sorts of conditions. Now, if you pay attention to the temperatures that we have here for methane and ethane, those are above the temperatures that you would need in order to have liquids on Pluto. Remember, I told you Pluto tends to be in temperature ranges of 30 to 60 Kelvin. We're still a little bit above this. So we wouldn't expect this sort of mixture to lead to liquid behavior there. So how are we going to tackle that? Well, there were lots of other molecules present. So what we can do is start thinking about all of those other molecules. What happens if we mix them together? What sorts of freezing point depression behavior do we get from those? I'm going to center my discussion on mixtures with nitrogen because nitrogen is the most common species on Pluto. And so you can kind of imagine that we have nitrogen floating around. Chances are we could enrich in one of these other species and maybe we could get the right concoction that leads to liquids being able to form. So I have the pure melting point information here in the second column. I have the depression that our thermodynamic model predicts in the third column. And so you can see that if you mix nitrogen with methane, you get a 14 Kelvin depression in the melting point, which results in a lowest melting point of about 48 Kelvin. If you do this with a bunch of other molecules that are known to be common, methane and carbon monoxide jump out as ones that start to get into that range where we know the temperatures of Pluto reach somewhat regularly. And so possibly there could be liquids forming from these mixtures. There are a number of other molecules that are interesting, and there's a whole lot of chemistry happening, but a lot of the more complicated molecules end up resulting in higher melting points. So them alone would not explain what we're seeing. There's a catch though. So carbon monoxide and methane look interesting, but the thermodynamic model that we've developed, it hinges on something in thermodynamics called ideal solution behavior. There's a lot on this slide. Hopefully I can break it down so it's not too complicated. What the ideal solution approximation states is that we are assuming that two different molecules interact with each other in the same way that they would interact with themselves. So if you imagine some molecule A, it has interactions with itself that have some magnitude, which is meant to be symbolized by this idea of UAA. If you allow it to interact with B, the ideal approximation says that those are essentially exactly the same, which gives rise to the model that we have. Two molecules of different types are never going to interact with each other in exactly the same way. So the next kind of component of this project is looking into how do molecules actually interact with each other in order to quantify how good or bad of an approximation is this. It turns out that testing it is not difficult. We can do this by just making these mixtures up and looking at what results from it. And so, for example, just to give you some numbers to attach to this idea of ideal behavior, if you take half a liter of methane, half a liter of ethane, mix them together, the resulting liquid of those two things would have a volume of 0.99 liters. So they don't add up exactly 1 to 1 in order to give you 1 liter. That deviation is this idea of an ideal solution. If you take water and ethanol, a really common non-ideal solution, if you mix these together, you get 0.96. So you can end up with deviations that end up being big enough that if you were able to put these into a glass, you would actually see the difference from pulling in half of one thing, half of another thing. They don't add up to what you might expect. This deviation of these values from what you might expect going into it, is oftentimes called the excess volume. And I'm going to use that word a little bit again as we think about quantifying this. So how am I going to test this? Well, my expertise is in a computational technique called molecular dynamics. And this hinges on this idea that if we take atoms, study how they move in their natural environments, we can use that to understand the world we see. So there's this great quote from Feynman: all of the things around us, I'm not actually reading it obviously, all the things around us can be understood in terms of the wiggling and jiggling of the atoms that make up those things. So we're going to take that idea to the limit. And so this oftentimes gets called the computational microscope. So on the right side of the slide here is my dog Monty. You could think about Monty's behavior being a product of the emergent behavior of the molecules that are buried at these deep scales below all of the emergent structures below that. So at some fundamental level, there are just molecules moving around that make up his behavior. Those molecules organize into larger sized proteins and structures like that, which come together to form cell membranes and stuff like that, all the way up to cells and organelles, all the way up to produce this dog. So what my group will do, what we oftentimes do, is look at that molecular scale structure in order to hopefully extrapolate up to the larger scales in order to understand the sorts of phenomena that we're interested in. In this case, we're interested in mixing these molecules together and seeing how ideal do they actually behave. And so in order to do that, we use this method called molecular dynamics. The details aren't too important, but it's not that complicated. So I wanted to give you a sense of what it actually is. If you remember fundamental physics, Newton's second law tells us that the force that an atom feels is related to the acceleration. And what we do is we just build up an understanding of that force on every atom. From that, we can predict accelerations. And then we just do that over and over again. We obsessively calculate this equation down here towards the bottom, where we can update what our position of an atom looks like by iteratively doing this repeatedly. And we do it millions and millions of times. The product of this is something that essentially looks like a molecular scale movie of all of our atoms moving around doing what they want to do. And from that, we can extract things like what are the densities of these solutions. And that is exactly the sort of information we need to test this ideal approximation. And so here's an example of one of these movies. A student of mine, Shai Dustrud, did this a couple years ago. And this was a simulation of methane. And here you see that it goes through a phase transition where it actually goes from the liquid phase into the solid phase. So we're going to be doing simulations very similar to this of methane, absolutely, and ethane as well. And we're going to look at them pure. We're going to look at them mixed together in order to understand, are these solutions ideal? And so here's an example of one of those methane and ethane mixtures. And if we take this and run it, we can extract from it that excess volume ideal. And so here you can see that when they get mixed together, they do not fall right on zero. If there was a straight line where that black dashed line was, that would be ideal behavior. The fact that it dips down is indicative that we are seeing real behavior manifest in these sorts of systems. It turns out, though, that methane and ethane are fairly ideal. So the deviation is not very large. If you were to do this for ethanol and water, you would see a much larger deviation. So we are able to quantify the real effects in this system, and we find that they are relatively small, which is justification for why the thermodynamic model that I introduced before agreed so well with the experimental results. So we have real behavior, but it's not that large in this case. You can do experiments to do this. Remember, in order to probe the methane-ethane system, we have to go all the way down to near 70 Kelvin and even below it. So these experiments are non-trivial. It is more than just taking these two things and mixing them in a graduated cylinder and something that you can do on the benchtop. You actually have to have pretty sophisticated apparatus. But if you do those experiments, you end up getting the same sort of trend that we predict. So the black dots here go in the same direction as our theoretical results in blue. And they're of the same order of magnitude. They're obviously not as clean, but remember, we have to forgive the experimentalists here. It's a really challenging experiment to do. Okay, so if we go through and do this for these mixtures that we're interested in, we can see that there are deviations from ideality. I quantified this in two different ways. One way we did it is by looking at the maximum excess volume that we observed. I just took the magnitude of it, so positive or negative. I just looked for deviations from ideality. And you can see the methane deviates a little bit in this metric, and carbon monoxide deviates even more. We've also been trying to come up with some more sophisticated ways of measuring this, and I wanted to put it on here because a student of mine has done a lot of work on this, which I'm calling so far the Ideality score. It's still a little bit sophisticated, a little too sophisticated and hard to interpret, but I think we're on a path where we might be able to actually take the information from this and feed it back into the thermodynamic model, which is kind of where we're trying to push with it. But you can see that these generally agree with each other and give some indication of the quality of these two predictions that we have. Okay. So I told you I wanted to weave together a few different stories. So here we're at the end of the first story. We're searching for liquids. We have found, I think, is a plausible explanation for how we could possibly see liquids on the surface of Pluto. If we mix these together, we can start to see depressions in the melting points that we observe of those mixtures. And these seem like possible situations where you could have liquids occurring. I want to transition to a different part of the solar system. In my title, I promised lakes and rivers. So I want to go somewhere where we know there are liquids and think about what's happening in those liquids that might be relevant to the formation of life. So on Pluto, this is still speculative. If we go to Saturn's moon Titan, so here is a composite image of that, we find that there are persistent lakes on Titan. And so these are places where we could actually think about what are the chemistries happening here, how are those relevant to what might be happening with prebiotic, pre-life biological-like molecules. So Titan is interesting because it is the only place in the solar system that we absolutely know there are persisting liquids on its surface. So we have liquids that are definitely there. The lakes there are made up primarily of nitrogen, methane, ethane, so similar sorts of molecules to what we were thinking about when we were talking about the surface of Pluto. But we're in a temperature pressure range where these end up staying liquids in the right places all of the time. And you actually get full cycles of behavior. So similar to what water does on the surface of Earth, methane does something very similar on the surface of Pluto, where it forms these lakes. It can evaporate, it can form clouds, it can rain down. You get rivers of this flowing across, you get erosive action from it. So we have a really interesting analog for the types of behavior that we see on Earth. And a lot of people think of Titan as kind of a early Earth analog that we can use to understand what might have had to happen on Earth in order to form life, maybe at a different place in the solar system. What I'm particularly interested in is thinking in these lakes, what do complex molecules do? So nitrogen, methane, ethane are not the things that we usually think of as forming life. But if we start allowing other molecules to integrate themselves into these lakes, what are they doing? Can we find little pockets of interesting places where chemistries could be happening that lead to maybe the sorts of processes that result in the kinds of molecules we are interested in? So leading to the formation of proteins and DNA and those things. It turns out that lots of molecules have been observed on the surface of Titan and other places in the outer solar system, and many of these have been implicated in the formation of biological molecules. So I had a student this last summer. We spent some time going through the literature, looking at molecules that are there, comparing that to lists of molecules that are important for the formation of biological molecules. And we came up with this list of molecules that kind of jumps through different categories. So some of them are hydrophobic, some of them are hydrophilic, some of them are known to react in certain ways that are interesting. And we decided to put many of these into models of these Titan lakes to see what would happen. And when we did it, we started from a totally random mixture, which is what you see on the bottom left. We went through this process of running a molecular dynamics simulation, like what I introduced you to a few slides ago. And we saw for many of them that they form little clusters. So in green here, I'm showing the formation of a cluster of one of these interesting complex molecules. And so what I want to do is look at which molecules form these clusters, and then a little bit of information about the clusters themselves. The way that I want to characterize these is with something called the radius of gyration. This is just overly complicated physics characterization of bubbles. So the radius of gyration allows us to look at whether or not a bubble has formed and what size is it. And if you look down this list of molecules that we've looked at, many of them form a bubble. Acetonitrile, benzene, hydrogen cyanide, all of these you can see are forming bubbles, and they are on this order of about 10 angstroms. That's an interesting size because it's about the size of a molecule. So we're seeing these things really tightly clustered together with each other, and they really like to be separated from the surrounding Titan lake environment. So these are prime locations for thinking about the sorts of chemistries that might be interesting for the formation of even larger molecules that might be relevant for life. We did find a few molecules that don't seem to go through this sort of separation process. Carbon monoxide and cyanoacetylene do not phase separate in the same sort of way, which doesn't mean that they couldn't be involved in these processes, but they're not going to separate and form a little region where we're ready for some interesting process to happen right away. So in concluding this section, in addition to finding liquids, we now have a set of molecules that actually come together in order to form little regions where we can think about interesting chemistries happening that might be relevant for the development of biological analogs, other places in the solar system. Okay, there's one last story that I would like to talk about. And this one is going to be taking molecules that are proposed to actually be prebiotic molecules and see how they behave. A lot of our motivation, and to be honest, a lot of motivation in this prebiotic field in general comes from some really classic experiments that were done back in the late 1950s, the Miller-Urey experiments. And what they did was they mixed together molecules that are known to be common in these types of abiotic situations, so very similar to Titan or Pluto, and they just sparked them and then looked at what fell out. And so if you take things like water and methane and mix them together and add a lot of energy to it, you get all kinds of molecules that start forming. It would be really cool if just DNA fell out of that, or if protein started falling out of that. But those molecules are pretty complex, and there are a lot of steps to get there. So what we've been spending a lot of time the last 70 years, and obviously even before these experiments, trying to understand is how do you get from these really harsh kind of random chemical environments to the really organized environments that life tends to produce. And so if you do these Miller-Urey experiments, you can get lots of interesting things that come out. I've only focused on the nucleic acids here, or nucleic acid analogs, because I want to spend a little bit of time talking about the behavior of those. And the literature is full of examples of these molecules and what they might be doing. And in particular, what people are interested in is how can you form them? So given that you mix these things together, given that you expose them to lots of energy, you see these molecules pop out. And so I don't even really want to talk about all of these other than just to make the point that we don't really understand the path that you need to go through in order to form something like DNA, something as complex and sophisticated as DNA. But we have lots of proposals out there. And so what my group has done, which is a little distinct from a lot of the other groups where other people have just been looking at what molecules can you form, what we decided to do is take some of these molecules and see if they actually behave like the DNA, the nucleic acid molecules that we're used to. And in particular, we chose to highlight one of them here. And so on the left, you see uracil and cytosine. Those are nucleic acids that biology as we know it uses to pass genetic information on. Those actually do not form too easily under these sorts of random conditions. You don't usually see those molecules. You do see the molecules on the right though. And if you look at these closely, you'll see that everything's the same, but there's a little OH added to it. For some reason, the addition of that OH causes this to be a more stable molecule and allows it to just emerge spontaneously out of experiments like the Miller-Urey experiment. And so we decided to focus on this molecule, or these molecules, and see, do they actually behave like the nucleic acids that we are familiar with? So can we take these ones that we know form under these sorts of prebiotic conditions, and can we see life-like behavior come out of them? One other thing that's interesting, or a couple other things that are interesting about these molecules that I want to highlight, is first of all, these molecules became quite, I don't know, popular, maybe not the right word, but they became a little bit more aware to the scientific community, because taking meteorites that have crashed on Earth and cracking them open and looking what's there, you actually find these molecules. So we have, beyond things like the Miller-Urey experiment, which is kind of an indirect measure of what you might see in proto-Earth types environments, we also have direct evidence that these things have formed in space and they actually are out there. So that's interesting. And then in addition to that, if you take them and just start mixing them together and allowing them to kind of go through reactions, they will actually spontaneously form biopolymers. What's interesting about that is if you think about a lot of the molecules that are important for life, DNA, amino acid chains that form proteins, they are biopolymers like this. So they form these very, very long chains of connected subunits that end up forming gigantic molecules that end up being the basis for a lot of the machinery of life. So the ability of these molecules to spontaneously form these long chains is indicative maybe of another kind of distinct analog to the types of molecules behavior that we see on Earth. And so what we decided to do was again use that same technique of molecular dynamics and probe the behavior of some of these molecules. And so what we did was we mixed them together randomly in water, and we just started to see what happened. And so this is very similar to the setup that I described in the previous section. And we're going to hopefully see something emerge out of this that will give us some insight into whether or not these molecules seem like they are similar to biological molecules. So here I've removed the water, and you can see that they're randomly arranged around. And so now the question is, what are they going to do if we just start following them along? If we just set up a camera and let them do what they do, will they do anything that's illustrative of life? It turns out that they do. Similar to the molecules that we saw in the previous section, they really like to clump with each other. And they will form these little clumps very quickly, and there does seem to be structure to them. And in fact, if you start analyzing that structure, we see hydrogen bonds emerge. If you know much about DNA, you might recall that a part of what's special about DNA is it has this base pairing process where different bases will come and stick to each other through hydrogen bonds. And this ends up being a really important aspect of how DNA performs the function that it performs in biology. We do not see anything as clean and concerted as DNA base pairing, but the fact that it exists seems kind of suggestive of maybe similar sorts of processes in very different molecules. So I'm going to call this tantalizing, even if it's not exactly the same. In addition to looking at this hydrogen bonding, we can look at that same radius of gyration process, or radius of gyration characteristic. And what's interesting about this is oftentimes biological molecules will take on smaller structures than they would if they were just totally random and flopping around. So smaller radius of gyration values oftentimes correspond to folding or forming kind of well-conserved structures that are important for the operation of whatever these molecules might be doing in some biological context. So if we go through calculating this size for these molecules, and then in particular, we correlate this with the number of hydrogen bonds, we actually do see pretty good correlation. This is not driven by what's happening with the water. If you looked at the leftmost plot, we don't really see a strong correlation between hydrogen bonds with water and the size that this thing takes on. But if you look at the intermolecular hydrogen bonds, so between pairs of these nucleic acids, or if you look at the hydrogen bonds that they form within themselves, when we look at these chains, we see a strong correlation with the size that they take on. So hydrogen bonding ends up with them collapsing and just taking on smaller structures. It's maybe a stretch to call this a direct analog for what DNA does, but it is similar in many ways, which suggests that maybe this could be a molecule that you could think about being kind of a transitionary step to getting to the sorts of nucleic acids we're used to. There's one last thing, well, there are many other things, but one last thing that I want to highlight in our analysis of these structures. One thing, if you spend a while looking at the structure of DNA, is that the base pairs stack with each other. So on the left, we have a picture of a DNA strand, and you can see these lines through it that almost look like a ladder. These ladders are formed from the pairing of nucleic acids with each other and also the stacking of them on top of each other. And what's interesting is if you spend a while looking at our simulations, we see things that look somewhat similar to this. So on the right, you see a snapshot from one of our simulations, and you can see 4 of our nucleic acid analogs stacked on top of each other in something that looks somewhat like the sorts of behavior that you see on the left. Now, life is very controlled. It has a backbone to these molecules. It puts a lot of effort into making sure that they do this. We are seeing a similar sort of behavior emerge out of a much simpler system that has some just kind of coarse similarities to it. And so a student of mine spent some time actually quantifying this and seeing are they really statistically stacked with each other, or did we just happen to find the right snapshot that makes it kind of look like that? And so he came up with a really nice, really robust way of quantifying this. And I'm going to show you a picture of it, and it doesn't look like much. And when he first showed me this, I was kind of disappointed. But if you dig into this, it actually ends up being really interesting. And so what we're looking at is how far apart those nucleic acids are, and then how likely they are to be stacked on top of each other. And buried at the far right side of this is a really intense peak that corresponds to stacking. Now, if you look at the whole plot here and zoom in away from that stacking behavior, so the C here, I'm not sure why my student chose C for the closest interaction, but he did. The C stacking here indicates that really strong propensity for nucleic acids of these forms to stack. And then if you look one away, you see the A spot, which corresponds to the next level of stacking. And so we actually see these kind of elongated structures, these multiple molecules coming together, forming these stacked-like structures that look like the sorts of base pair stacking that we see in DNA, without all of the machinery that life has put together in order to make it happen that way. In addition to that, we found that we also have this kind of this rotated stacking that occurs. So B here is an indicative of where molecules are rotated from each other and kind of approach each other from the side. So it looks like the backbone and all the machinery that life has put together to cause DNA to fit together in the right way is important, because if you don't have that, you'll get this kind of misalignment approach that also ends up being a dominating feature in these sorts of similar but obviously distinct systems. Okay, so we looked at one set of molecules that are very similar to biological DNA. These things form spontaneously and they polymerize, so there's a lot about them that is interesting for understanding where the molecules of life might come from. And we found that they have a number of features that are very similar to DNA. So they hydrogen bond with each other, they have these base stacking processes, they form these tight clumps that seem like interesting places where you could imagine them arranging themselves just right in order to go on to do the interesting complexities of life. There are obviously features that are not so lifelike. Intramolecular hydrogen bonding seems to be preferred over something similar to base pairing, the intermolecular process that we see in DNA. But I think there's something interesting here that is suggestive of maybe a path that would be interesting to look at. Okay, that is the set of stories that I wanted to tell you. The first thing I want to do is thank the VPR's office and Andy Kopish for inviting me, for all of you to taking time to listen. But there are a number of students who actually did the work. So Kevin Gotchenauer is the one who did the nucleotide work, a lot of that at the end that I talked about. There are a number of students that have done the thermodynamic modeling, the molecular dynamics, and even some of the experiments that I presented today. So Shai, Lavinia, Anna, and Alex are those students. And then all of this work largely rose out of collaborations that I've had with other people kind of inspiring me, bringing conundrums that have burst out of the work they've been doing. So in particular, Jennifer Hanley and Will Grundy are the planetary scientists that have been involved with collecting a lot of this data. They brought me a lot of these problems, and it's been an opportunity for me to get involved with it. And I guess the last thing I'll think is I've been fortunate to get funding from a number of sources, but in particular, NASA has given us a couple of grants that have allowed us to really spend some time thinking about this and working on it. And with that, I would be happy to take any questions that you might have.
Wonderful, Garrick. Thank you. That was amazing content. We have a number of questions flowing in, which is good. People are engaged. The first one came a little while ago. It's regarding your first story when you were talking about Pluto. But could Pluto's low atmospheric pressure impact whether something is liquid or solid? And do you simulate that? Does a machine at NAU lower the pressure?
That's a great question. It could have an effect. The pressure has less of an effect than the temperature. So you would anticipate it being most important to target the right temperatures, and then the pressure is oftentimes secondary to that. We can simulate that. We can set arbitrary pressures in what we do in the simulation, and they also can modify that in the experimental setup that they're doing. So we are very aware of that, and we do incorporate it into the models and experiments that we're doing. But again, it tends to be less of an effect than temperature. So there's not quite as many interesting stories that usually emerge out of that.
Okay, that was good. I understood that. That means it was good. Yeah, a couple more flowing in here. Next one is, do enantiomeric preferences, or EP, emerge in the simulations? Do we expect EP on other planets? And are there atmospheric signatures detectable, such as polarization?
Great question. Another great question. So one of the really interesting things about life is that if you look at, so my two hands, right and left hand, they are similar to each other, but they are mirror images, right? It turns out that a lot of molecules have a similar sort of behavior where I can take the same atoms, put them together, and they will be mirror images of each other depending on what way you put them together. It turns out that life is almost entirely constructed out of molecules that are right-handed, so to speak. So on Earth, you have the possibility of both right and left-handed, but life is built out of just one of those. You do not see that sort of preference out of the molecules I'm talking about. When they form through things like the Miller-Urey experiment, or the types of experiments that led to, well, processes that led to the molecules that we observed in those meteorites, for example, we don't see that sort of handedness emerge yet. So it seems like that must be something that occurs at some subsequent step outside of the sorts of processes that we've imagined so far. That's really one of the great conundrums and one of the huge biological signatures that you would think about for life. Life needs one of these versus the other one because it needs the repetition, the structure that emerges out of that order, but we're not quite sure where it comes from.
Okay. And the follow-up, more of an opinion on that, but why do you think Earth displays the same EP?
I think it becomes about, we're going a little bit beyond my expertise, but I'll speculate here. I think it comes about because proteins and DNA need to have very well conserved structures in order to behave the way they need to for life to happen. If you have mixtures of those two things, of those two right and left-handed structures, you get molecules that look very different. They have the same bonding with each other, but they end up behaving very differently. And so by controlling to have just one of those, life has made it even more controlled when you think about protein structure, for example. So I think it was an emergence out of the control that you need to have for these assemblies to behave the way they need to have life.
Especially intelligent life.
Hopefully, yeah.
Yeah. Okay, next one. The Miller-Urey experiment uses a spark like lightning to investigate the beginnings of life. Does Pluto or Titan have any processes which make sparks? Although it sounds like you may not need a spark.
So there have been, so my direct answer is I'm not actually sure. I don't know if there's something quite like lightning on the surface of Pluto. But as an addition to that, the spark is nice because it produces a lot really quick. So the Miller-Urey experiment is something you can do quickly and you'll get all kinds of gunk that falls out of it. You don't have to have anything that dramatic. You have cosmic rays and rays from the sun hitting Pluto. Those are generating these more complicated molecules. Those continue to go through reactions with each other to form even more complicated molecules. So we don't necessarily need that spark of lightning if we have time to wait for those high energy events to occur. And there have been other experiments that have followed the Miller-Urey experiments using different sources of energy that have seen the same sorts of molecules fall out. So absolutely, the Miller-Urey experiment is kind of this analog for what happens with lightning, but there are other processes that you can imagine giving rise to similar sorts of complex molecules.
Okay, and a couple more here.
Great.
One comes from Frank. How much extra stabilization do you think you would get if you could include the actual electronic pi-pi overlap? And do you think that plays an important role in stabilizing the stacked structures in a way that leads to biomolecules?
I do think it would be important. So the types of models we're using do not handle that sort of pi-pi stacking in the rich way that we know real molecules do. And that would be something fascinating to look at. I think you would probably see more complexity to the sorts of structures that we get out of it if you had a more rich potential energy surface to look at. But we haven't looked at it yet.
Okay. And one more here from Mary. If you could direct a space probe to gather more data to enlighten these investigations for you, where would you send it, and what would it look for?
I don't think I'm creative enough to answer that question. I will parlay to the Dragonfly mission that is approved by NASA and is going to happen. I'm really excited for that one. So this is a probe that's going to go to Titan, and it's going to collect new information. It's going to give us more information about what's happening on Titan. So I think there's going to be all kinds of wonderful data that comes out of that we can use to critique our models, that will make our models even more useful as we can contrast the information we're getting from them with information that comes from the Dragonfly mission. So I think there's going to be a whole new opportunity to understand these, really exotic places and kind of imagine ourselves there, what it means to have life forming potentially there.
Okay. And another one just trickled in. Let's see. Does the Dragonfly mission have a lander?
I have no idea. Sorry.
I don't know that one either. And I actually have a personal question just from my own rudimentary understanding of the habitable zone. It sounds like if we're detecting water and ice in regions and bodies that are outside of the habitable zone, do you ever think that they would revise how large that zone is?
I think the habitable zone makes a lot of sense when you're thinking of Earth analogs. If you think of life needing water in the way that we know life on Earth needs it, then the habitable zone kind of gives you some constraints in order to do that search for Earth-like planets. I guess part of the argument that I'm making here is that it's interesting to expand a little bit beyond that, because if you go to these other places, there are other types of chemistry that can emerge. And so, for example, one of the molecules in my second story that we looked at is a molecule that we know forms in the interstellar medium. We also know that it forms on Titan. That molecule is way too reactive to occur on Earth. If it forms in the upper atmosphere, it very quickly will bump into something else, go through reactions, and disappear. But that molecule is known to be a part of pathways that lead to biological-like molecules. So it's kind of intriguing to think about these other environments that present kind of different reactivity where maybe molecules can form. And there's a pretty kind of robust amount of evidence that maybe some of the molecules that life needs actually initially formed in other places and ended up being deposited on Earth. And so this could be a pathway for that to happen. Some of the things that life needs ended up forming somewhere else. They somehow got spun off into space and landed here and were able to do whatever they needed to do to form life. But those other environments were important potentially for them coming together the way they did. So I think kind of thinking beyond our traditional water-based way of life originating gives us kind of new avenues to form molecules that you wouldn't think of if you're always thinking of an aqueous environment.
Okay. We had an attendee, Dan, raise your hand. Dan, if you have a question, please type it into the Q&A box. You can see that at the bottom. And while Dan is doing that, Garrick, I'm going to kind of start our wrapping up. I'm going to share my own screen, if you don't mind, so that we can show the people some of the supporters of the festival this year. And I'm going to give a couple more minutes for Dan to maybe give a question if he has one. And oh, here it comes. Nope, not yet. Anyway, Garrick, I really appreciate you coming to speak tonight. This was awesome. I work at Lowell Observatory, so it's really nice for me to see this kind of community involvement. And let's see here. Giving a couple more minutes for Dan to get his question in, but might not have an opportunity to. So we are pretty much out of time. If anybody could take a look at the people who make this a free activity and a free sponsor. Garrick, we hope to have you in future festivals, giving many, many more speaks, giving many more talks like this, because it's really informative and it's helpful for other people in the community to be able to know and understand what's being talked about in what you do.
Absolutely. Thank you. It's all been wonderful. Yeah, thanks.
You got it. Okay, everybody. Thanks so much for joining us this evening. And if you would have